Dear students, today we'll have a lecture number 3 about the medical protozoology. As you remember, this is the part of medical parasitology during which we can study the parasites who belong to the subkingdom Protozoa. And today let's discuss the part number 1. But at the beginning, look at this picture where you can find all diversity of protozoa parasites. They belong to the empire Cellulata, subempire Eukaryota, kingdom Protista, subkingdom Protozoa. And there are 3 main phylums: Sarcomastigophora, Ciliophora, and Sporozoa or Apicomplexa, inside which we have majority of parasites. Who they are? To the Sarcomastigophora belong dysenteric amoeba, Leishmania, Trypanosome, Trichomonas, and Giardia, majority of parasitic species. To the phylum Ciliophora belongs Balantidium coli, only one parasite, and phylum Sporozoa includes famous malaria parasite and Toxoplasma gondii. About each of them we will discuss in the frame of our lectures. Today we'll speak more closely about dysenteric amoeba and Balantidium coli. But before to speak about them, let's discuss the general information about the protozoa in whole. Who they are? They are unicellular creatures. We have very special organs of locomotion. They may be pseudopodia, flagellums, and cilia. Pseudopodia, temporary outgrowths of cytoplasm, we can see at the example of dysenteric amoeba. Flagellums, organoids in the form of long thin threads which are placed at the anterior part of body, you can see in the case of Giardia intestinalis, Trichomonas hominis, Leishmania, and Trypanosoma species. And Balantidium coli is covered by cilia, organoids in the form of numerous short threads. That is why pseudopodia, flagellums, and cilia, there are 3 types of organs for locomotion among protozoa. How the protozoa can feed themselves? What are the ways of nutrition? Those protozoa who consume solid particulars of food, they can use the mechanism of phagocytosis, and they are able to form food vehicles around the food substances. For example, here you can see formation of food vehicles step by step. Amoeba can feed with the solid food particles. But many parasitic species, contrary to free-living ones, are surrounded by fluid-like food, and that is why they can use pinocytosis for nutrition, and they can form pinocytotic vehicles. Some protozoa must use the special organs of excretion, especially those who can live in the media with the fresh water. They are able to use contractile vehicles to eject extra water. That is why these contractile vehicles are used as the organs of osmotic regulation. Plus to water, it is possible to excrete the products of dissimilation, as well as it is possible to accumulate of oxygen from water using the cell membrane as well as these contractile vehicles. At the picture you can see very simply organized contractile vehicle of amoeba and very complicated contractile vehicles in the case of Paramecium caudatum, infusorian shoes, well-known infusorian. Here you can see two vehicles, each of which must have small canals and the central reservoir, and the pore through which the water may be ejected from the body. What about the reproduction? Protozoa can use two ways of reproduction, asexual and sexual. Asexual reproduction may be presented as binary fission, very simple way, when, thanks to mitosis, one maternal cell can divide into two identical daughter cells. The same story you can see among dysenteric amoeba, Balantidium coli, Leishmania, Trypanosoma, and majority of protozoa. But some of them can use multiple fission with the special name schizogony. Multiple fission occurs very interesting. Firstly, inside the maternal cell, we can see increasing by number of nuclei thanks to multiple mitosis. And to the end of these divisions, we can see formation of one cell with many nuclei. This cell is well known as schizont. And finally, around each cell, we can see separation of cytoplasm and formation of daughter cells under the name merozoites. Such multiple fission we can see in the life cycle of malaria parasite, different Plasmodium species. Some protozoa are able to use the sexual way of reproduction. For example, some infusorians from the phylum Ciliaphore and the malaria parasite. Also, inside the body of female of Anopheles mosquito, we can see the sexual reproduction. What is the most important characteristic of this way? Sexual reproduction is not so typical because it occurs by exchange of nuclear material between two individuals. Here you can see the conjugation of infusorian with transmission of parts of micronuclei between these two creatures. And as a result, after conjugation, we can receive two individuals, but with mix of heritable material inside the nuclei. And after this conjugation, we can see the separation of two individuals and they undergo to the normal binary fission. Altogether, conjugation and binary fission can be well known as sexual reproduction among unicellular creatures. It is not so typical sexual reproduction, we can't see males and females. Only in the case of malaria parasites, we can see male gametocytes and female gametocytes formation, but about this way of reproduction, we can speak a little bit later. As usually, generally, in life cycle of protozoa, we can see two stages. One stage is non-active under the name cyst, and second stage, very active under the name trophozoid. Trophozoid is able to move, to feed, and to reproduce. But all this it can provide when the environmental conditions are very favorable. When the conditions seems to be not so favorable to survive, trophozoid surrounded with the cyst wall, the special envelope, and transforms to the cyst. Also, cyst may be used for penetration into the host in the case of parasites, or can be distributed with the aid of water or wind for the great territories. That is why cyst is dormant stage, trophozoite is active stage. Excistation is the process by which from the cyst comes out new trophozoite. And firstly, we can speak few words about amoebiasis, disease which is provoked by dysenteric amoeba, parasite from the phylum Sarcomastigophora, class Sarcodina, order Amebina, the dysenteric amoeba. This disease was described since 1875. A lot of people which can live in the area with warm, humid climate, tropical or subtropical, can suffer by this parasite, dysenteric amoeba. At this picture you can see the picture and photo of cyst stage and few photos of trophozoite stage, small vegetative form and large vegetative form. And now let's speak about a little bit about life cycle. Life cycle is started by penetration of mature cyst into the mouth of person. This way is peroral way, through the mouth. The mature cyst with 4 nuclei can go into our large intestine. And here you can see excistation, when the trophozoite comes out from the cyst. And this trophozoite is well known as small vegetative form. It can live inside lumen of large intestine of human, can't provoke any harm because it can consume different particulars of food, different bacteria which are very numerous in the human's large intestine. And when this forma minuta, small vegetative form, goes to the rectum, transforms to the precyst and to the cyst, and with the feces of patient, they can go to environment. This situation is well known as cyst carriage. By cyst carriage, human can stay healthy, but it can be very dangerous for all other people who surround him because he can spray the dangerous invasive stage, the cysts, and other people also can start to be sick. Cyst carriage, this is a situation when the immunity system of human is rather strong and can suppress the development of parasites inside. But occasionally, and nobody knows exactly, what change of conditions can provoke the change of this life cycle. It may be change of climate, when the human can move from native country to other country with untypical for him climate, starvation, different disease can provoke terrible transformation, and small vegetative form will transform into the tissue form. This tissue form starts to consume the mucosa of large intestine. Really, instead of simply swim in the lumen, they can start to consume the mucosa. They can provoke formation of ulcers, bleeding, and appearance of very strong diarrhea with blood and mucosa in the feces. That is why tissue form is harmful trophozoid of dysenteric amoeba. Appearance of many erythrocytes in the lumen of intestine can provoke next transformation. As the quantity of small vegetative forms are able to transform to large vegetative forms under the name forma magna. And this forma magna is able to consume erythrocytes which they can find in the lumen of intestine. That is why forma magna is not harmful, it just appears as the reflection of appearance tissue form, the harmful form. In the case when I can see tissue form is the mucosa of large intestine and large vegetative form in the lumen of large intestine, I can see intestinal amoebiasis disease, intestinal amoebiasis. But sometimes tissue form are able to go into the blood vascular system and go into the liver, lungs, heart, and even the brain. This situation is well known as extraintestinal amoebiasis. Therefore, we have three situations: cyst carriage, intestinal amoebiasis, and extraintestinal amoebiasis. More light is cyst carriage, after that intestinal amoebiasis with the symptoms like diarrhea, and after that extraintestinal amoebiasis like the great problems for the health. How the cysts are able to go to the persons? As usually it happens, where we have poor level of sanitation, and when the water may be contaminated with human's feces. And drinking water will transform to the dirty water. And of course, when we can take the food by dirty hands and consume unwashed fruits and vegetables. And one more very important thing is when we can drink the drinks with ice cubes, in the case when ice cubes are made by the fresh unboiled water, possibly with the cysts of dysenteric amoeba. That is why prevention of this disease will be all methods linked with personal and public hygiene, and of course reduction of exposure to fecal matter containing cysts. Because there are no vaccination and no prophylactic drugs against amoebiasis. And one more time, in the life cycle of dysenteric amoeba and amoeba histolytica, we can see only one host, and this is only human. The cyst is invasive stage. It can go to our body perorally through the mouth. This parasite can affect humans' digestive tract, especially large intestine. Inside our intestine, this parasite can multiply asexually. And the cyst can come out from the human's body with the feces. And that is why when we can make the analysis of fresh stool specimen, when we can find here the cysts, it will be like Conclusion: human is cyst carrier. But when I can take the fresh feces from the patient and I'm able to observe inside the specimen only the large vegetative forms, forma magna, this is the sign human is sick with intestinal amoebiasis during acute period, where the symptoms are bright, and I need to organize the treatment for such person. To understand successful or not the treatment is, I can take one more time the feces for analysis, and when I can find more cysts than forma magna, it can show for me that disease can undergo to the hidden stage, dormant stage, like the chronic amoebiasis, chronic intestinal amoebiasis, and I have to continue the treatment till the end, when inside the analysis I will never observe single cyst. What it will be like the end of amoebiasis in person. But some patients don't want to go to doctor, to address to doctor when the situation will be not so dangerous, when diarrhea finished, abdominal pain finished, and all return back to normal situation, and they can interrupt the treatment. As a result, amoebiasis can undergo to the cyst carriage or to the chronic situation, to the chronic duration. And such people may be in danger because the disease can return back. Each moment, small vegetative forms, forma minuta, will be able to transform back to tissue forms and will start to consume mucosa of large intestine. That is why you can read more attentively the ways by which the cysts can be spread from person to person, and you can see what happened by extraintestinal amoebiasis, when tissue form can be spread by bloodstream and can affect different organs. For your consideration here, in short, the description of intestinal amoebiasis, the most important features and symptoms. And here I want to present for you some photos in which you can see different stages of dysenteric amoeba life cycle. Tissue forms. They are here staying in the mucosa of large intestine. And this is result of their activity. You can see formation of ulcer and epithelium and some mucosa of large intestine may be destroyed. This process can provoke bleeding and presence a lot of red blood cells in the lumen of large intestine, and immediately other small vegetative forms will transform to large vegetative forms, forma magna. You can see their photos here. There are few forma magna which are able to consume erythrocytes. And inside the cell you can see a lot of food vehicles. Inside each of them can stay erythrocyte. And please remember, tissue form is harmful because it is guilty for formation of ulcers and for bleeding and for diarrhea and abdominal pain. But forma magna can't provoke any harm. They just can eat red blood cells inside intestine. They can't provoke some problems in humans. But especially in the stool specimen, I can see forma magna, like reflection of presence tissue form inside the mucosa. tissue form never can be found in the human feces specimen, only forma magna. Of course, intestinal amoebiasis can bring a lot of problems for the patient. Firstly, perianal skin damage, which can be provoked by the diarrhea, and we can see formation of ulcers at the internal area, internal surface of large intestine. All these ulcers provoked by activities of tissue forms. And here you can see some of these amoeb can stay at the surface of large intestine. But extraintestinal amoebiasis can be found not so often, but here you can see the scheme how with the bloodstream tissue forms can affect different organs, and more often we can see the liver who is affected. And in the liver are formed liver abscesses. Here you can see some pictures which are made with the aid of cadaver material and with ultrasonography and MRT. And you can see these lesions, these inflammations in the tissues of liver and increasing of liver and spleen by size. Sometimes by extraintestinal amoebiasis you can see formation of such lesions with addition of secondary infection, which can provoke very severe situation by the patient and sometimes the death of patients. Together with dysenteric amoeb, inside human's intestine we can find a lot of different species of amoeb, but they are non-pathogenic. They can use the same large intestine, but just to find the food. They are not parasitic species, but possibly they may be confused with dangerous dysenteric amoeb, Entamoeba histolytica. And more often it's happened with Entamoeba coli, famous intestinal amoeba. Entamoeba coli can also be found in the lumen of large intestine, but it is able to differ the cyst of Entamoeba coli from the cyst of harmful Entamoeba histolytica. Inside the cyst of Entamoeba coli, I can see 8 nuclei, not 4 but 8. But sometimes when the doctor have not great experience to use the microscope by this identification, it can make mistake by calculation of number of nuclei, and instead of 8 it can calculate 4 and make the prescription that this human suffered with dysenteric amoeba. But truly it's just Entamoeba coli intestinal amoeba, nothing harmful. That is why doctor must be very attentive by establishing of diagnosis. Except dysenteric amoeba, I want to speak with you about some other species of amoeba which also are dangerous for the health, but we will not discuss them during our lessons. And number one here is Acanthamoeba. This amoeba, which membrane must have the spines, that is why we have this name Acanthamoeba, can be spread in different freshwater natural reservoirs, and they can affect the humans. They can fall into the humans through the eye, through the nasal passages, or sometimes through the broken skin, when the human can wash with unboiled fresh water in which this amoeba may be found. And more dangerous this amoeba is for those persons who can apply optical lenses, because this amoeba is able to penetrate and to stay between cornea and lens and can destroy the cornea of eye, can provoke the blindness. You can see inflammation of eye like the consequence of activity of this amoeba, and the final, when we have loss of vision and formation of great atrophy of cornea. And one more species who is also dangerous and also can be found in the fresh water is Nigleria fowleri. This amoeba is amazing because it is able to transform not only into the cyst stage, but into the stage with flagellums. Really, it can swim in water like flagellate stage, and it is able to penetrate into the humans, which can swim in such water pools like amoeba-like stage, and can transform into the cyst also. People who can swim in such water are in danger because this Nigleria, this amoeba, is able to go through the olfactory nerve and olfactory bulb directly to the brain and can provoke very fast inflammation of the brain tissues and even the death of patient. And such situation can occur so quick that even doctors don't understand the reason why it happened, and only autopsy can show appearance of such amoeba sitting inside the nervous tissue of the brain. That is why when you can swim in the water pool, you can do it very carefully and don't take the water into your mouth, or possibly don't breathe with the water. Be careful. And for today, at last, we can discuss the story about other parasite who belongs to the phylum Ciliaphora, Balantidium coli. In this picture you can see trophozoite and the cyst. Trophozoite of Balantidium coli is covered with cilia and it has two nuclei. Macronucleus, the big nucleus, and micronucleus, the small one. In the cytoplasm we can see a lot of food vehicles and few contractile vehicles. And except all this, trophozoite of Balantidium must have the cellular mouth and cellular throat, peristome and cytostome, and even cytopharynx, with the aid of which they can take a food. Inside the cyst you can find just macronucleus and very well developed cyst wall. This parasite, Balantidium coli, can provoke the disease under the name balantidiasis. It can be spread everywhere where humans can raise the pigs, because pigs are natural reservoir hosts for this parasite. Balantidium coli can be found in the large intestine of the pigs. And they can't provoke any harm for pig, but when human can receive the cyst stage, which is invasive stage, to the mouth, as usually with the unwashed vegetables or with the dirty water, the cyst can come into the large intestine and transform there into the trophozoite. In the case of Balantidium, we have also two situations, cyst carriage and the disease balantidiasis. By cyst carriage, inside the intestine of person, we can see just trophozoite, which can sit and live here without harm, which is able to transform into the cyst in the case when it can go into the rectum. In the feces, it can transform to the cyst. And such person is healthy but can be the source of invasion for other people. For example, for the family members. But occasionally, trophozoites can be activated, can transform to the harmful trophozoite who will start to consume mucosa of large intestine. And we can see the disease balantidiasis in this case. But I can tell for you that human's immunity can suppress this parasite rather active, and balantidiasis is not so dangerous disease as amoebiasis. And that is why human can suffer with abdominal pain and diarrhea, but it can be finished by themselves, and human don't want address to doctor because they can suppose that maybe it's something wrong with the food, not something wrong with the parasites. At the first part, the humans who can fall sick with balantidiasis, they are the farm workers which can raise the pigs, which can take care about the pigs, and they can receive such parasite from the pig's feces when the people forget about personal individual hygiene. That is why here the diagnosis of cyst carriage is made by observation cysts in the feces, but acute period of balantidiasis with the symptoms can be identified by presence of trophozoites in the feces. And please draw your attention that in the case of Balantidium coli, we have only trophozoite without different division. No small vegetative form, no tissue form, no large vegetative form, only trophozoite. Trophozoite who can live normally, quietly, and trophozoite who is able to consume mucosa of large intestinal wall. And that is why, as prevention, it will be don't forget about rules of personal hygiene, because this is fecal oral transmission. Yes, when the human feces with the cyst can go with the contact of the vegetables and water, and other people can be sick. And especially the special veterinarian observation of the pigs, which can be found at the farms. It seems to be each pig must be observed for the presence of such parasite, and in the case when this parasite will be observed, we have to give the treatment for the pig, because main source of invasion for people will be the pigs. And after that, the human who is cyst carrier. For today, it's all about these two big parasites, dysenteric amoeba and Balantidium coli. See you later. During our next lecture, we will discuss the story about the protozoa with the flagellums, about Leishmania species, Trypanosoma, Trichomonas, and Giardia intestinalis. Goodbye.